My name is Ed Chapman, and this is our second video cast talking about the cell cycle. And we finished up last time talking about the phases of mitosis: prophase, prometaphase, metaphase, anaphase, and telophase. Today we're going to start out talking a little bit more closely about the mitotic spindle. Now, the mitotic spindle is an array of microtubules that grow and develop inside of a dividing cell, and their job is to move the chromosomes around and to change the shape of the cell to facilitate the whole process of a cell dividing. Now, there are two parts of the mitotic spindle. The first part are the kinetochore microtubules, which here I've labeled with these green arrows, and they are the darker lines here. Now notice that the kinetochore microtubules are actually connecting to the centromeres in the chromosomes here. So, for example, this kinetochore microtubule here is starting here at the centrosome, it's shooting out, and it's attaching to this chromatid of this chromosome on this side. Non-kinetochore microtubules are the other part of the mitotic spindle that don't attach to the chromosomes. Okay, they just slide past each other, and they're going to be involved kind of like a bracing system to fill out the mitotic spindle. Okay, now to understand how this works, scientists had the basic question: what is causing the chromosomes to move? And once they discovered a way of dyeing the mitotic spindle with fluorescent dyes, they were able to make these microtubules show up, and they immediately wanted to know, like, which end of the microtubules are actually getting shorter, because these kinetochore microtubules have to be shortening if they're going to be moving these chromatids towards the poles. So there's two ways they can shorten. They're either being shortened at this end, down here near the centrosome, which is kind of like a fishing rod reeling in a line to pull these chromosomes this way, or they're being shortened up here at the kinetochore end. And if it's being shortened there, it's kind of like the chromatids acting like a little Pac-Man and kind of eating at it and following it down this way. So they were trying to figure out which way, and of course, an experiment is the way you do it. What they did is they labeled the microtubule, the kinetochore microtubule here, using a laser. And we'll put a little dot right here. Now, if this microtubule is shortening from this end, this segment here should get shorter, and the dot should move this way. If it's shortening on this side, then the dot should move this way. And the track marks the center, okay? And you allow the cell to go through anaphase, and the data shows that the mark moves towards the kinetochore. And I'll just kind of animate it here. So that proved that you actually have the Pac-Man theory here winning. The kinetochore microtubules are being taken apart or depolymerized here at the kinetochore end. Okay, so you can kind of think of it as like little Pac-Man chewing at it this way. And as they shorten it from this end, each of the chromatids then pulls apart and moves to opposite poles. All right. Plants and animals go through cytokinesis in different ways. If you remember, plant cells have a cell wall that gets in the way. So the way a plant cell is going to go through cytokinesis, it's going to build something called a cell plate between the two new nuclei at the end of telophase. And what happens is Golgi apparatus in the cytoplasm are producing vesicles, which are fusing to build a wall right down the middle here. And this wall then becomes the middle lamella, and then each cell builds its cell wall on the opposite side. So we end up with a triple layer here of protection between these two new plant cells. Now, animal cells do it differently. Animal cells actually are able to pinch in, kind of like somebody tying a string around a water balloon. And this is called the cleavage furrow. It's actually produced by microtubules or microfilaments on the inside of the cell membrane shortening in the middle, kind of like a girdle or a belt, and it pulls in, separating the two dividing cells during animal cell cytokinesis. But what about bacteria? Well, bacteria don't go through mitosis or meiosis or cytokinesis. They have their own process. They only have one chromosome, otherwise known as a nucleoid. They have no organelles to worry about, and their division is a lot simpler. It's called binary fission. And all that happens is their nucleoid, or their single loop of genetic material here, duplicates or replicates. Each replicated copy is pulled apart by some mechanism, probably due to attachment to a lengthening cell membrane here, though I'm not sure. And then the cell pinches in the middle, much like we saw in animal cells with the cleavage furrow. And finally, you end up with two new bacterial cells, each one a perfect copy of the original. No worries here, very simple process. Okay, now, cell division has to be regulated. Okay, You can't be normal without regulation. If you think about it, growth and development are complicated processes. They have to happen right. Each step has to happen with the right timing, or you get irregularities. Okay, so that means there must be some timing and some control going on here. I like to think of it as the cell cycle. You get it? Bicycle here, cycle, must have a driver. So what is the driver? What is the thing that controls the cell cycle or regulates it? Well, the regulation is happening at the molecular level, which means it's being regulated by molecules. Okay, scientists asked the original question: does each event in the cell cycle, like G1, S, G2, prophase, metaphase, anaphase, does each one trigger the one after it? Does the cell somehow know that it's finished one step so it knows to move on to the next step? No, that isn't the way it works. What scientists have figured out is there are chemicals in the cytoplasm that actually regulate the cell cycle. And these signals, remember we talked about cell signaling? These signals act like stop and go signals, and they're called, very creatively, cyclins. These cyclins are able to activate certain kinases, and the cyclin and kinase complex is then able to regulate the cell cycle. Okay, an example of this would be signals called growth factors. They're in the cytoplasm and they're released and they trigger a cell to start dividing. For example, there are some growth factors that are released by platelets. So let's say you get a cut on your hand. The cut causes the oxygen exposure of the tissues to oxygen in the atmosphere triggers platelets to release growth factors, which then switch on cell division. And the cells around the wound then start to divide and seal the wound and heal it. The healing process is triggered by growth factors. Now, cells in a culture, and this is a culture box or something kind of like a petri plate here, if we put some cells in here, cells are inhibited by density, which means crowded cells stop dividing. So if you put a few cells into some nutrient material in a box and you left them alone and kept them at the right temperature and everything, they're going to grow and divide until they form a single layer on the bottom of the box, and then they're going to stop. And if you scoop some out, okay, they'll divide sort of like a wound here to fill the gap. But crowding stops cell division, and scientists, of course, want to know why this is. And what they figured out is that it's all about the concentration of growth factors and nutrients in this group of cells. If the growth factor concentration and the nutrient level concentration falls too low because of the crowding, the cells stop dividing. They kind of know there's not enough room, there's too many cells, there's not enough food, there's not enough signaling going on here. Everybody stop dividing. Okay, if you take some out, they're able to recognize, yeah, there's some space, there's some extra nutrients, there's some extra growth factors here. Everybody, or the ones at least close by here, start dividing to fill the gap. It kind of makes sense. So cell division is switched off by crowding in normal tissues. Some cells, most of the cells in the human body, for example, are anchorage dependent, which means that if they're free floating off by themselves, they don't divide. They have to touch something. All right? They must touch a surface or a substrate and attach. And once they do this, the attachment signals the cell to start dividing. Now, this takes us to cancer. Cancer is a disease of cell division. Cancer cells, in some way or another, have escaped cell cycle controls. They're able to ignore the signals that normally would tell normal non-cancerous cells to stop dividing. For some reason, they're able to ignore the fact that they're running out of growth factors. They don't respond to crowding, and cancer cells kill by strangling, choking, and invading normal tissues and organs. This inspired ancient Greeks to name the disease that we call cancer. They named it after the Greek word for crab, which is cancer. All right? Kind of a side note here, there was a lady lived back in the 1920s named Henrietta Lacks, and unfortunately she had a cancer. And doctors were able to remove her cancerous tumor, and they saved some cells from it. And they found out that her cells were able to grow in petri plates very easily. They never got tired, they never stopped growing, they just kept growing and growing and growing. So her cells actually founded a line of cells that are immortal. They're used in cancer and cell division research to this day, and they were named after her. They're called HeLa cells. Capital H, little e, capital L, little a. HeLa cells, named after Henrietta Lacks. Now, normal cells can convert into cancerous cells in a process called transformation. And Immune cells normally recognize cells that have transformed and destroy them, or send them a signal to die. And cells transform because they've either had a gene turned on or induced, or another gene turned off or repressed. So scientists have found that there's basically two sets of genes involved in the transformation of normal cells into cancerous cells. And these genes are called oncogenes. You're either born with them, or they're introduced to your cells by not organisms, but vectors that can carry genes from one organism to another, viruses. All right, two big cancer words you need to know about: benign versus malignant. Frequently hear these words on medical shows and things. A benign tumor is simply a tumor that's growing but not spreading throughout the body. On the other hand, malignant tumors are tumors that start releasing cells that float off in the bloodstream or in the lymph system and settle in new places and cause new tumors to form. And when that happens, we say that the tumor has metastasized or gone through a process called metastasis. Okay, so these cells are travelers, so they're called metastatic cells. And this is a bad thing. That's why it's always easier to cure cancer if it's caught early and the tumor is removed before it has a chance to metastasize. There are many questions about cancer that are unanswered to this day because we really don't understand all the ins and outs of regular cell division in normal cells. So maybe one day you guys will become oncologists and contribute research to this burgeoning field. There's a lot of work that needs to be done to solve the cancer problem. It seems like everybody eventually gets some kind of cancer somewhere if you just live long enough, and people are living longer now. Okay, hope you've enjoyed this video cast. Hope it helps you understand how the cell cycle takes place and how it is regulated. We'll stop there.